Thank you so much, Krista, for that introduction, and thank you so much for the Planning Committee for inviting me here. I really appreciate the opportunity to give this talk. So as Krista said, today I'll be discussing the sources of cognitive variability in phenylketonuria, or PKU, beyond Phe. Here are my disclosures and our objectives for today. So first I'm going to start by explaining the pathophysiological mechanisms contributing to neurocognitive outcomes in patients with PKU. Then we will discuss the impact of Phe management on PKU pathophysiology and the predictive value for neurocognitive outcomes. Then we'll evaluate other factors beside Phe levels, which might be predicting neurocognitive health in these patients and how they can optimize treatment. And finally, we'll assess how clinical research findings can be applied to clinical practice to ultimately improve patient outcomes. So as many of us know, PKU is an inherited metabolic disorder of amino acid metabolism, which involves a mutation in the PAH gene, which encodes the phenylalanine hydroxylase enzyme. And this enzyme is predominantly expressed in the liver, and it hydroxylates the essential amino acid phenylalanine, or Phe, into tyrosine. And in PKU, this hydroxylation reaction is inhibited due to a deficiency of the PAH enzyme or dysfunctional enzyme activity, depending on the severity of the condition for each individual. And this ultimately leads to elevated Phe levels in the blood, usually above 360 micromoles per liter, which, as you can see, is much higher than the Phe level in healthy individuals, which is usually around 60 micromoles per liter. And if left untreated, the consequences of elevated Phe can be devastating. It can lead to severe intellectual disability, stunted growth, hyperactivity, and mood disorders. The image here reflects the historical observations before the 1960s, in which children who were diagnosed beyond the newborn screen were severely affected, as you can see in the boy on the left. His sister, who is on the right, appears healthy, given she was diagnosed by newborn screening and she received treatment during the critical periods of growth and development. And while late diagnosis and treatment lead to the symptomology I just described, early treatment with poor adherence can also lead to a range of cognitive deficits which are a product of underlying structural defects, neurophysiological dysfunction, and abnormal myelination. And while it's clear that there are many paths by which elevated Phe can contribute to these changes, the direct mechanisms have still not been fully elucidated. So for the next several slides, I'll review the hypothesized mechanisms whereby Phe toxicity directly contributes to the neurocognitive impairment or plays an important mediating role. So to start, excess Phe reduces the concentrations of other large neutral amino acids in the brain due to the transport competition at the blood-brain barrier. Each of these large neutral amino acids uses the LAT1 transporter, and the LAT1 transporter has the highest affinity for Phe. So when blood Phe levels are high, LAT1 gets saturated with Phe and it blocks the entry of those other large neutral amino acids. This ultimately leads to a decreased availability of those essential amino acids, which decreases cerebral protein synthesis, and that affects the concentration of important enzymes like tyrosine hydroxylase, shown as TH here, or tryptophan hydroxylase, shown as TPH. And what's not shown here is that can also affect some really important receptors, such as the glutamate receptor. This decreased availability of large neutral amino acids, and specifically of tyrosine and tryptophan, can also impair the synthesis of the important neurotransmitters dopamine and serotonin. And deficiencies of these important brain messengers create microstructural changes by decreasing synaptic plasticity and axonal growth. Excess Phe during acute or chronic exposure can also disrupt glutamatergic transmission. Glutamate is the major excitatory neurotransmitter in the brain, meaning that it stimulates cells into action. And it works by binding to the glutamate receptor on nerve cells, which then triggers a change or an action in the cell, and it allows the communication signal to continue to be transmitted. This fast signal and processing of information is ultimately critical for learning and memory. And evidence from rat and mouse studies have demonstrated 3 different pathways through which acute Phe elevations can depress glutamatergic transition at both the pre- and the postsynaptic sites. The first pathway, which you can see here, let me try this out, in this image on the left, and it's labeled with a 1, in which Phe can actually compete with the glycine binding site of one of the glutamate receptors called NMDA. And glycine is an agonist for these receptors, so this can depress the activity of this important receptor. The second pathway, labeled with a 2, is that Phe can actually compete with the glutamate binding site of non-NMDA receptors, such as your kainate and your AMPA receptors. And the third pathway is that Phe can attenuate the presynaptic release of glutamate. And long-lasting Phe exposure not only perpetuates these mechanisms, which we see with acute Phe exposure, high Phe exposure, but it can also create some compensatory changes in glutamatergic synaptic transmission, such as increased glutamate from the presynaptic area, increased aging of those specific receptors, and then also increased density of those glutamate receptors. Epigenetic changes also represent another very important mechanism through which elevated Phe affects neurocognitive outcomes. Research in this area is generally limited, but one human study by Dabrowski et al. Provides some very nice evidence to start considering this important mechanism. In this study that I show here, methylation patterns were compared in the leukocytes from 5 therapeutically non-compliant, as determined by Phe greater than 1,200 micromoles, and 5 therapeutically controlled individuals, as denoted by Phe less than or equal to 360. And the differential methylation in these disease groups was identified by comparing them to 7 healthy controls, and then the patterns were compared between these 2 groups. And what we can see immediately is that there was aberrant methylation in both protein-coding genes and non-coding RNA genes. And within those protein-coding genes on the left here, we can see in those circles that it's actually quite rare to see any hypermethylation in both of the groups, both the compliant and non-compliant groups. However, hypomethylation seems to be a little bit more common, but we don't really see a difference between the compliant and non-compliant PKU patients. In contrast, in the non-coding RNA genes, we see that differential methylation was quite extensive, but that's really only in the therapeutically non-compliant patients. Beyond this analysis, there was also an additional analysis not shown here in 2 brains from PKU patients, which showed that several of these genes that were changed also led to expression changes in genes that were really important for myelination, neurotransmitter regulation, synaptic support, synaptic function, axon integrity, and mitochondrial mobility. Another important mechanism to discuss is oxidative stress, which is driven by the overproduction of reactive oxygen species, which I'll refer to as ROS. And at low levels, these unstable molecules actually play a very important role in redox signaling, meaning that they help with the activation of various cellular cascades. Excess Phe, however, can disrupt redox signaling and ultimately cause damage in proteins, lipids, and DNA. And it can do this through 2 different important mechanisms. First, by affecting the primary sources of these unstable molecules, and namely the electron transport chain, the transmembrane carrier nicotinamide adenine dinucleotide phosphate, it's a mouthful, NOx, or nitric oxide synthase, NOS. ROS levels can be further increased by a decrease in both enzymatic and non-enzymatic antioxidants. And several micronutrients play a very important role as non-enzymatic antioxidants or as cofactors for these enzymatic antioxidants. And for this reason, the Phe-restricted diet also plays an important role in this mechanism. Phe can also inhibit energy metabolism and dysregulate calcium homeostasis. And Phe can inhibit the activity of a very important enzyme, pyruvate kinase, in the glycolytic pathway, which converts phosphoenolpyruvate to pyruvate. And for calcium metabolism, Phe can modulate this plasma membrane calcium ATPase, leading to a greater extrusion of calcium from those neural cells. Excess Phe also, of course, impairs myelination. Myelination, as a reminder, is the process by which myelin sheaths, which are produced by oligodendrocytes, both surround and isolate the axons, and ultimately supporting the saltatory conduction of action potentials, or the spread of information. And hypomyelination is characterized by a decrease of myelin production, and demyelination arises from damage to the myelin. There are several different mechanisms by which Phe can actually affect both of these processes. The first involves reduced activity of enzymes involved in the biosynthesis of important myelin precursors, which you can see here, such as cholesterol, sulfatides, and ceramides. Secondly, Phe can decrease the rate of synthesis, as we've already discussed, of several cerebral proteins. And in this case, for myelin, some that may be important are the neural cell molecule adhesion, or myelin basic proteins. The third contributing mechanism is that aberrant DNA methylation, which we talked about as well, can also alter the transcription and the translation of myocillin, which is a protein expressed within the myelin sheath. And finally, Phe can also have direct cytotoxic effects, which can be exacerbated by oxidative stress, energy deficits, and inflammation. The resulting changes that we see with this myelination can lead to abnormalities in white matter, which are generally most evident in the T2 weighted images that we can get with an MRI. And we see those changes mostly in the paraventricular white matter, followed by the occipital, frontal, and temporal white matter regions. So when we consider the varied mechanisms that we just reviewed, the most critical factor in determining the neurocognitive impact is ultimately time. This nice figure was synthesized in a recent review paper by Ravelli and Longo from several different supporting articles. And what we can see here is that elevated Phe can affect every single phase of brain growth. During fetal life, which is on the left here, the effects on the developing brain by maternal PKU can result in microcephaly, neuronal loss, and corpus callosum hypoplasia. After birth, in the first couple of years of life, exposure to high Phe can lead to severe cognitive impairment, and in some cases epilepsy, and this can likely be due to the impairment in the formation of the dendritic spines and affected synaptogenesis. Then in later childhood, Phe can have a slightly different impact. It can change neurocognitive performance. It may be attributed or may be associated with ADHD and lead to changes in IQ. And this could be because it's affecting dendritic pruning and synaptic remodeling. And what connects all of these different phases is brain myelination, because that's something that can occur starting in fetal life through adulthood. And at any age, that can be impaired by hyperphenylalaninemia with consequent impairment of executive functions. So to prevent the neurotoxic effects associated with elevated Phe, it's recommended for patients to maintain Phe within the very narrow range of 120 to 360 micromoles per liter. And now there's several strategies in which we can accomplish this. The first and foremost is the steep restriction of dietary protein, and in its place, patients consume small amounts of naturally low protein foods like fruits and vegetables, in combination with specialized medical foods which contain all essential amino acids with the exception of Phe and are often fortified with other micronutrients. More recently, PKU management has also introduced 2 pharmacotherapies which are approved. And the first is sapropterin dihydrochloride, which can act as a synthetic cofactor. And patients who are on this treatment can generally have a more liberalized diet, but it's not a fully normalized diet. The other pharmacotherapy is pegvaliase, which is enzyme substitution therapy, and this treatment can allow patients to have a fully unrestricted diet once they respond. And despite early diagnosis and the use of these treatments which control Phe levels, individuals with PKU still exhibit neurological changes and cognitive deficits. This includes a small decrease in overall intellectual functioning and then more notable impairments in executive functions, including self-monitoring, planning, organization, emotional control, and working memory. This has been demonstrated now by numerous studies, but here I show a more recent study which was done in early treated individuals with PKU, both children and adults, and it utilized a well-established tool called the brief, which is a preferred tool because it captures executive function within the context of an individual's everyday life. And the two figures on this slide show several executive function domains on the x-axis here, gender and norm t-scores on the y-axis, with higher scores representing greater impairment in each of these areas. And what we can see is that relative to the average, which is shown with a t-score of 50 and a dotted line here, the two domains that had consistent impairment in both adults and children were working memory and something called MCI, which they made a metacognition index. And this index included working memory, planning, organizing, and task monitoring. And between these, working memory actually had the highest impairment in both children and adults. This study also demonstrated that Phe is not a consistent clinical predictor for cognitive outcomes. Here I show working memory as our outcome on the y-axis, given this domain had the highest level of impairment in the previous slide. And both figures here illustrate the correlation between average Phe levels over the past year and working memory t-score. And what we can see is that both children and adults have a very weak relationship between these two variables. And even when looking at a particular Phe level, we can see that there's a large range of working memory t-scores, suggesting that there's other exogenous factors that are independently contributing to this variability or modulating the neurotoxic effect of Phe. Complementary evidence has also been found in longitudinal studies. A good example of this is a study by Monti et al., which evaluated the quality of metabolic control on intra-individual variation in mental functioning in 65 early treated patients with PKU who had lifetime Phe data. In this sample, IQ was examined at two different time points during the first 2 decades of life. These two time points were about 10 years apart, with the mean age at time 1 being 10.2 years, and the mean age at time 2 about 20 years. And metabolic control during each of these 2 periods was computed as a mean of yearly median Phe levels. And what was interesting is they found some unexpected inconsistencies in their results. As highlighted in green here, we can see that IQ actually decreased in 26% of the patients who maintained good lifetime Phe control, as defined by having Phe less than 360 at time period 1 and Phe less than 600 at IQ time period 2. On the flip side, in patients who actually maintained poor lifetime Phe control, as highlighted here in red, IQ actually improved or kept stable in 72% of that sample. And beyond these inconsistencies, the study also found that only 16% of IQ variability could be attributed to metabolic control, meaning that 84% was coming from some individual unknown determinants. Inconsistencies in the relationship between Phe and cognition have not only been found in early treated patients, but also late diagnosed patients as well. Several unusual cases were compiled in a recent study here, which looked at 59 late diagnosed individuals with PKU who had plasma Phe levels greater than or equal to 1,200 micromole. And interestingly, all of these individuals, as you can see in yellow, sorry, it's a little hard to see, had intellectual functioning within the normal range. But it was interesting also to find that even though their IQ was normal, they still had other cerebral symptoms, indicating that there may be other paths or causes of these findings. And this study said that these unusual cases likely represent 10% of the PKU population. So there's a lot more work to do to understand what's causing this variability. So now that we've reviewed the mechanisms caused by Phe, I'm now going to take some time to think about the mechanisms that may be underlying the cognitive variability that is not caused by Phe. This is a nice schematic that was also found in that last paper, and it outlines some of the main hypotheses regarding the possible mechanisms that are causing this inter-individual variability. The first, as you can see here with the 1, is that there may be differences in the transport of Phe and other large neutral amino acids across the blood-brain barrier. The second is that there may be differences in transport within all the cells across each of those membranes for Phe and large neutral amino acids, or other mechanisms that are affecting those cells' functionality. And finally, number 3, that there may be a difference in the vulnerability of one or more of these intracerebral processes that we've discussed before. So I'll now explore the currently limited but growing evidence for each of these mechanisms. The first area that's been explored is our variants within the LAT1 transporter. And as a reminder, this is how Phe and other large neutral amino acids enter the brain. So one LAT1 variant listed here has been found to increase cerebral Phe concentrations. Evidence for this variant came from a 2021 study in which 20 patients with classical PKU from Poland who were 12 to 25 years of age were examined. And these patients were genotyped for this variant, and what they found is that 13 were carriers, 12 were heterozygous, and 1 was homozygous, and 15 were wild type. And the figures here represent a comparison of blood Phe on the left and brain Phe intensities on the right as measured by MRS between those carriers and wild type individuals. And on the left, we can see that blood Phe was slightly lower in carriers than wild type individuals, but that this difference was not statistically significant. In contrast, on the right, we can see that there's a higher intensity of brain Phe in the carriers of this LAT1 variant compared to the wild type individuals, and this was statistically significant. And while the exact mechanism for how this variant may actually create a functional change in LAT1 has not been determined at this time, it has been hypothesized to affect the expression of the gene, given it overlaps the binding site of a transcriptional repressor. Other relevant gene polymorphisms have also been identified in the SHAPE gene family. Members of the SHAPE protein family are scaffold proteins which localize to the postsynaptic density of dendritic spines in the excitatory synapses of the brain. And these SHAPE proteins are also involved in the organization of the glutamatergic synapse by linking the receptor proteins on the postsynaptic membrane to the actin cytoskeleton of the dendritic spine. Furthermore, it's also been shown that SHAPE proteins mediate the maturation of neuronal dendritic spines. So this is something that's also very important for the plasticity of the nervous system. A recent study suggests that these SHAPE variants may have a protective effect on cognitive development in PKU. This evidence comes from 4 untreated PKU patients with near normal cognitive outcomes. And next generation sequencing was used to find which other genes that these patients may have in common beside PAH. And in 3 out of the 4 of these patients, 3 variants were found in 3 genes of the SHAPE family: SHAPE1, SHAPE2, and SHANK3. And to understand a little bit more about these variants, this study used computational analysis to characterize the pathogenicity. And what they found was that all of these were quite benign and did not abolish the function of the SHANK protein. By further using some in silico modeling to see the effect of those variants on the structure of the protein, they did find that these changes created some post-translational modifications in the proline residues of the proline-rich domain of the SHANK protein, and that this could influence the glutamatergic synapses, cytoskeleton, and the cytoskeleton, and ultimately contribute to a more optimal synaptic density. So now going back to our schematic again to discuss another potential mechanism, which is nutriture. And by this I mean nutrient intakes and nutrient status, and particularly for those neurologically relevant nutrients. And given the central role of diet in PKU, there have been several papers which have examined nutrient status in this population. And there's been one previous meta-analysis by Para et al., which has specifically focused on nutrients that play important roles in brain development and function. And those nutrients are shown here in blue on the left. And in that list, I've bolded the couple of nutrients that had at least 4 observational or interventional studies in the literature, which have previously evaluated the status of these nutrients in individuals with PKU compared to controls. And in this study, meta-analysis methods were used separately for each of these nutrients to determine how these nutrient levels compared between PKU and controls. And what I've done is compile those standard mean differences here in a forest plot on the right. And what we can see is that there were no neurologically relevant nutrients in which status was significantly greater in PKU versus non-PKU individuals. However, there were 3 nutrients, cholesterol, EPA, and DHA, which had significantly lower status in PKU individuals compared to non-PKU individuals. And for the several nutrients that were not bolded in that list on the left, due to a paucity of data in the literature, we can also consider the other component of nutriture, which is intake. The data on this slide comes from a study in 2017, which did a very thorough dietary evaluation of PKU patients who were solely on diet therapy or on diet therapy and sapropterin. And here I've summarized again a subset of neurologically relevant nutrients. And in this graph, I show the percentage of patients who were not meeting the estimated average requirement, or EAR, or the adequate intake level, or AI. And we can see those that are not meeting that based on the whole diet in a dark gray bar, and those that were not meeting that based on natural food alone with the light gray bar. And now in blue, we can see all nutrients that were at least 10 to 50% of the sample were not meeting the EAR or the AI based on the whole contribution of the diet. And what we can see in the lighter blue bar is that this percentage greatly increases when solely thinking of natural food alone. So what this data emphasizes is that there is a high prevalence of suboptimal intake for nutrients that are critical for brain development and function. And another interesting component of this is that it really questions our assumption that a liberalized diet in those patients who are on sapropterin and potentially pegvaliase, that this may not actually improve the diet. So now we'll transition to talking just about a couple of nutrients that we've actually looked at the connection between the nutrient status or intake and cognitive outcomes. This is something that's actually quite rare in PKU literature. There's only been 2 nutrients where this has been done, the first being DHA and the next, which is choline. So DHA is quite abundant in the brain, and it functions as a core component of the plasma membrane. And it's involved in helping with the fluidity of that membrane and the function of it. And This table here compiles the 6 different controlled or randomized controlled trials that have looked at the impact of DHA supplements, fish oil supplements, or formulas fortified with DHA on neurocognitive outcomes. And what we can see in 2 of those 6 studies, one which was an RCT and one which is a controlled trial, was that DHA supplementation with doses of 15 milligrams per kilogram resulted in shorter P100 latencies. P100 latency is considered the most reliable clinical indicator for visual evoked potential, which is a widely used measure for neural maturation. And out of the 6, only 1 randomized controlled trial reported beneficial effects for motor skills and other neurocognitive functions such as dynamic balance and total score, but the others have inconclusive findings. And some of the reasons for this could be the heterogeneity in the interventions, using very different outcome measures, and finally, different lengths of intervention. The other nutrient which has been investigated by our group is choline, which has several important roles for the brain. It serves as a precursor for myelin, a precursor for phospholipids, and a precursor for the neurotransmitter acetylcholine. And we have done one study in collaboration with Sean Christ's group in Missouri, in which we did a remote study that included 40 early treated PKU patients and 40 demographically matched controls. And in this remote study, we collected several different things. We collected 2 different measures of metabolic control, one a recent Phe, and the other an index of dietary control, which we calculated based on lifetime Phe levels. And we also collected 3-day dietary records in order to assess total choline intake. And finally, as you can see on the right, we looked at several different cognitive scales related to working memory in order to develop a working memory composite. And with that information that we collected, we looked at the relationship between total choline intake and working memory outcomes. And just as a note for some brief demographics, choline intake ended up being quite low in this population at 255 milligrams, which is significantly below the recommended 425 for adult women and 550 milligrams for adult men. And what we can see in the figure on the right is that when we adjust for energy intake, intellectual ability, and childhood Phe levels, that we do see a significant relationship between total choline intake and working memory outcomes. But what is interesting is that this relationship gets attenuated as Phe levels start to increase. And what's interesting about this still is that in that large group of PKU patients that have been diagnosed early and treated continuously, choline may represent one aspect that is leading to some variability in their neurocognitive outcomes. Another important moderating factor is the psychological sequelae, and most notably anxiety-related disorders or anxiety symptomology. When we think of anxiety in PKU, we typically attribute that to poor Phe due to its relationship with those decreased neurotransmitter levels. But a recent study supports the importance of also considering the psychosocial burden of PKU and how that independently impacts anxiety and overall mental health in this population. This study by Aronoff et al. Used the illness intrusiveness scale in 25 females with PKU to evaluate the impact of PKU on various aspects of their lives. And you can see some of those various aspects here, anything from health, diet and work, to community and civic involvement. And this scale, just for a little bit of background on it, is a validated 13-item self-report survey, which has a total score ranging from 13 to 91, and then sub-scores for each of these subcomponents, which range from 1 to 7, 1 being that the component contributes very little to this illness being intrusive, and 7 that it contributes a lot. And the central hypothesis for this whole illness intrusiveness is that treatment factors and disease factors lead to this illness intrusiveness, which then affects subjective well-being. And what Aronoff's study found is that, first, that there was a total mean score of 44.85, which is really not that high, thinking through the total score of 91 as an option, but that they had these interesting subscores, one being health, which was rated quite high as a 4.75 in adults and 3.8 in adolescents. And when following up this survey with some subjective interviews, what this author found is that anxiety was something shared by every single one of the patients as a main component of their health. And in terms of the relationship between metabolic control and anxiety, we have some additional evidence that there's an inconsistent relationship with Phe, or that there may be other main causes of this, in that same remote study that I described for the choline intake. In this study, we also looked at an anxiety composite score, and this score was developed from 3 different anxiety measures, one which was a self-report, one which was an other report based on a SEBA, and finally one which was a structured neuropsychiatric interview based on the MINI. And the scores that came from these 3 different scales allowed us to create 2 different groups, one which was a high anxiety group and one which was a lower anxiety group. And here you can see the criteria for meeting high anxiety, which was having either criteria for that based on the neuropsychiatric interview or being considered at risk on either that self-report or other report metric. And what we can see in the graph on the right is that there was not a significant difference between Phe levels in individuals who were considered higher anxiety versus lower anxiety. This study also evaluated if anxiety moderated the relationship between recent Phe levels and working memory. So as described in the previous slide, working memory was developed from several subscores to make a composite. And what we can see in the figure here was that there was a negative relationship between Phe levels and working memory scores for individuals who had low anxiety. And those individuals are shown by that solid blue trend line or the filled in blue dots here. But post hoc comparisons also revealed that the performance of those lower anxiety individuals decreased as Phe levels increased relative to our controls, which are shown here in gray. This trend was different for individuals who had higher anxiety. For those individuals, as you see with a dotted trend line here, despite what their Phe levels were, their working memory did not change. It stayed below the control level. So now that we've talked about the couple of factors which may contribute to some cognitive variability in PKU, the question is, how do we begin to identify other contributing factors? So one suggestion, and I think this is more of we're all moving toward this approach, is combining many different omics technologies. This can include genomics, epigenetics, transcriptomics, proteomics, and metabolomics. And these methods can all be integrated together through correlation, the construction of different molecular networks, and machine learning to help us discover new biomarkers, create new mechanisms and understand new mechanisms that are leading to this phenotypic variability, and finally to identify therapeutic targets. So now to summarize, this has shown us that the pathophysiology and the phenotypic expression of PKU are very complex. Excess Phe during critical periods of neurological development increases the risk of cognitive impairment, but the threshold for this risk differs among patients. There have only been a couple of factors at this point which have been identified that impact or are associated with individual vulnerability to Phe or variability in neurocognitive outcomes. And as we discussed, those were some gene variants in LAT1 and the SHANK gene family, some neurologically relevant nutrients, and namely DHA and choline, and finally anxiety. And in order to truly develop a personalized treatment approach for PKU, it's very important that we consider and further investigate this heterogeneity. And finally, some implications for research and clinical practice. So I think in clinic, we tend to focus for PKU on getting filter papers, looking at Phe levels, but it's really important that we make nutrient and psychiatric assessment a priority, and that we do this more frequently with patients so that we can get a better handle on how these factors are impacting their overall health. And when looking at neurocognitive performance in research, I think it's very important that we also consider anxiety and other psychological conditions as key moderating and confounding factors. And in the clinic, again, when making decisions about a patient's diet, it's really important that we consider the full nutrient profile and not solely focus on protein for these patients, as there can be various other nutrients that we've discussed that have very significant roles in neurological development and function. And finally, it's a priority to think about factors that are affecting monitoring and rather than simply reflecting neurological damage or cognitive impairment that's already been done. And I want to give my acknowledgments to our team, the SERGG Metabolic Nutrition and M&T for P group, and then our external collaborators on some of that work with choline and anxiety. Thank you so much.